and welcome to today's webinar. I'm Louisa Diva, and this webinar series is brought to you from the Capitalise project. The aim of these webinars is to promote communication and collaboration across the photosynthesis community, and today we're reaching far and wide to Australia. We're delighted to welcome two high-profile plant scientists, Professor Bob Furbank from Australian National University in Canberra and Dr. Maria Ermakova from Monash University in Melbourne. Okay, so our first speaker today is Professor Bob Furbank. He's an internationally renowned plant scientist that takes a systems biology approach. His focus is on improvement of photosynthetic performance, carbon partitioning and yield in C3 and C4 plants, and as well as gene discovery and field performance. His research spans synthetic biology, metabolic engineering, and global remote sensing and phenomics of crops. He's a node lead researcher in the C4 Rice Consortium and has led both the ARC Centre for Excellence for Translational Photosynthesis and the High Resolution Plant Phenotyping Facility at Canberra. So today I'm going to pass the camera over to you, Bob, and tell us about your future approaches to improving photosynthesis in crops.
Thank you, Louisa. It's a particular pleasure to be here because I've known Louisa since her PhD days, and she was a leading light in having produced some of the first mutants in Amaranthus in knocking out some of the C4 enzymes, in particular pepcarboxylase. So yeah, it's great to be here, and I will do a double act today with Maria, or Masha Ermakova as we call her. So I hope everyone can see my screen. Is that all working, Louisa?
That's looking good.
Yeah, excellent. Okay, so today's a double act. What Masha and I will be doing is to give you a bit of an update on some of the work that's been going on in our Centre of Excellence for Translational Photosynthesis, which has much the same goals as your EU program, and that is to bring our basic strategic photosynthesis research into a crop improvement context. And we've come to the end of a 7-year funding period for this centre, so it's quite opportune for Louisa to give us the opportunity to speak on this. So the title I gave Louisa was Improving Crop Photosynthesis, Genomes, Phenomes and Synthetic Biology, but Marsha will cover most of the synthetic biology aspects with her talk on C4 photosynthetic improvement, and I'll give more of a general introduction which covers more of the genomics, phenomics approaches that we've been using in the Centre of Excellence, and I'll focus on a couple of key areas that we've been successful in. So I think everyone is, I guess, a bit bored with seeing the same slide of the rate of genetic improvement of crop yields over the last 2 decades, where we're going to be by 2050, and where we need to be by 2050 to feed 1 billion people. So I stopped showing that slide, and I show instead this beautiful Broidel painting from the 16th century, which shows people bringing in the wheat crop. And while that doesn't really give us an indication of how we're tracking with providing enough food for our burgeoning population, the following statement that goes along with it does, and that is, oops. Now, here we go, that's right. In the next 50 years, we'll have to produce more food than we've consumed in the history of humankind. And that was a statement by the Chief Executive Officer of CSIRO back in 2009 when we were opening up our phenomics facility. And I guess, while that's a bit of a shock jock statement, it really, for the general public, it means more than a series of graphs and saying we have to produce 70% more food than we currently are. But the problem with that kind of statement is that it pushes everything out into the future. And this is one of the problems we've had with funding for climate change research. Everyone said, oh, well, you know, 2050, that's years away, don't need to worry about that. But what we've seen over the last few years is that the food security crisis is here right now, and we've seen that with the effects of climate change induced extreme weather events with supply chain issues to do with COVID. We're in a situation now where, unlike the people of the 16th century, where if you had a severe weather event and a crop loss, the village immediately starved that season, what we perceive is that food prices go up. We go to the supermarket, we pay more. We see on television the effects of famine in sub-Saharan Africa, but we don't have that strong connection with our food system. But I guess what I want to say with this slide is that the problem is pertinent to our current situation, not to the future, and we need to really act now as researchers to contribute towards solving this grand challenge, a global grand challenge. And we need to keep in mind that even with current technologies of high throughput genomics and crop breeding, it takes 7 to 10 years to produce a new wheat variety, for example, from first cross to a finished product that the farmer can grow in their field. So looking back in history is pretty instructive as a bit of background activity to see where we've come from in terms of improving crop yields and where we need to go in terms of our future emphasis, and why photosynthesis has become such a key trait target for crop breeders. This is a little slide. The top left-hand corner is an Australian wheat grower back in 1903. And as we can see, the wheat crop at that stage is above his head. The bottom left-hand corner is what a wheat crop looks like today, where wheat's barely come up to our knees. And the reason for this is the introduction of the GA-insensitive dwarfing genes by flagship breeders, not like the Nobel Prize winner Norman Borlaug. And the strategy here was to reduce the stature of the crop, to change the carbon allocation so that less carbon was invested in the unproductive stem and straw, and more carbon was invested in the grain. So these dwarfing genes allowed us to progressively, over the last 30 years, increase grain number markedly and improve harvest index, which is the proportion of the crop which is harvestable grain. So this shows basically where we've gone with our progressive breeding over the years. And the introduction of the dwarfing genes produced almost a doubling of crop yield on a per hectare basis. But what we've seen subsequently is that we've reached an asymptote with improving grain number and harvest index. And we basically cannot reduce the structural biomass of the crop any further without reducing the photosynthetic push. And what we're finding now, both in rice and in wheat, is that improvements in yield potential gain are becoming limited by photosynthesis, not by grain number. So step changes in grain number, for example, just result in a large number of unfilled grain. So where do we go? Well, obviously we need to improve photosynthesis. So what we've seen as a consequence is that the tables are kind of turned. I'm used to, in my career, going to crop breeders and multinational companies and saying, look, we need to improve photosynthesis. This is what we can do for you. And since 1999 in rice, the rice growers have been, and breeders have been coming to us and saying, how can we improve photosynthesis? And about a decade later, the realization that we were becoming source limited or photosynthesis limited in wheat also became apparent to the breeders, which resulted in the beginnings of the Wheat Yield Consortium and then the current International Wheat Yield Partnership. So over the last decade, really, we've seen a burgeoning group of activities addressing this issue of how we can improve photosynthetic efficiency and productivity in our major crop plants, ranging from major Gates-funded initiatives like the C4 Rice initiative, originally started at Erie, the RIPE initiative led out of Illinois, which when we add up the dollars spent by the Gates Foundation, we're looking at over $100 million investment. Then we're looking at a whole range of different activities across our major crops to look at improving photosynthesis, both by more traditional crop breeding approaches, but also by synthetic biology and GM. And that's where our Centre of Excellence came in. So in 2013, in Australia, we invested about $37 million in a 7-year project across 4 universities and 3 partner organisations aimed at bringing our basic strategic research into that crop improvement arena. and move from model systems, as Louisa said, out into our major food crops. So what kind of approaches why am I excited about this? Well, I think the future is exciting for us because for many years I had a lot of difficulty to try and convince crop breeders that our basic strategic discoveries in photosynthesis, which really are, at 60 years worth of really high quality biochemistry and physiology globally, how they could be of use to them in their breeding systems. Now with genomic selection and the combination of high throughput genotyping and genome resequence, combined with our high throughput phenomics tools that have been developing over the past decade, I think we've got a real opportunity to translate our research into new crop varieties, and this is at a level which we've never really seen before. So we're seeing more and more large germplasm panels of our major crops, and the image in the top left-hand corner is a diversity panel, global diversity panel of sorghum, which was held in Queensland by our one of our partners, Dave Jordan and co. These panels are either have high density markers across them, or the genomes have been resequenced so that we can really map the genetic blueprint of the phenotypic characters of those plants if we have the capacity to analyse many thousands and thousands of lines for their phenotypic performance. And now we can actually do that with some of our high throughput phenomics tools. So we've got two approaches. One is phenome to genome. We can take these panels of plants, examine their performance from a photosynthetic perspective, and then look at the genetic architecture that underpins those traits. And then that can be used directly by identifying superior material for crossing into target environments, quantitative trait loci for more traditional marker-assisted selection. But importantly, breeders now are interested in the complete genetic blueprint, not just a few anonymous markers. So we can use our knowledge of allelic variation and how gene sequence varies across these entries in germplasm collections for our photosynthetic improvement work. The other direction we can come from is we can use our 50 years of experience to mine candidate genes in those genomes of these vast array of diversity sets. And we can look for allelic variants, which vary in, say, gene sequence for the coding regions of our major Calvin cycle enzymes or our electron transport components, and look at how those allelic variants map to photosynthetic performance and use this information for increased genetic gain in our breeding programs. Whoops. So why does that particularly excite me? I mean, that's not a new thing. I guess what's improved over the last decade is our phenomics tools and our genomics tools. But bringing this together with synthetic biology, I think, gives us a chance of getting to where we need to go in terms of improving genetic gain in our cereal crops to meet food security demands. And by using the approach that I've talked about on genome to phenome or phenome to genome, we can generate gene candidates that we can use in synthetic biology approaches with things like CRISPR-Cas enabled gene editing. We now have easy transformation in the majority of our food crops. So we can actually find variation in genes that we can see as important for photosynthetic performance. And we can make mimics of that sequence in elite breeding material and rapidly move that into the breeding system. So this is really in its infancy, but I think it offers a huge potential for us globally to use our photosynthetic know-how for crop improvement. So a combination of digital crop breeding and synthetic biology and genome engineering, I think, is the answer, and you'll hear a bit about those two things today. So no doubt, you're aware of much of the work that's been going on across RIPE. You've heard of some of the translational photosynthesis work, no doubt, and read about it, and also the C4I SCATES program. Much of this work has been delivered through synthetic biology and GM approaches. We have a vast array of different targets that we've identified from our years and years of research, looking at what the limiting steps are in photosynthesis. And these comprise a whole range of enzymes that we know are limiting under certain environmental conditions to carbon fixation and also to the light reactions. And While we're only now starting to see these move from model systems like tobacco and Arabidopsis and into crops like wheat and rice, we're beginning to see gains. And at the bottom there are some of the papers that have come out of the RIPE Consortium and other researchers addressing these sort of synthetic biology approaches. But also the model-assisted guide to what we should use as our filter, if you like. What are the best candidate genes? In the center of excellence and also in RIPE, we've used this approach of using systems modeling of plant performance to scale from the enzyme to the leaf, to the crop, to the field, to yield, to aid us in thinking and prioritizing what our activities should be. So exploiting natural variation is what I'm going to focus on in the next 10 minutes or so. And I think anyone who's done a simulation versus CO2 concentration curve knows how long it takes to do a good LICOR experiment. We need to let plants equilibrate in the machine. It can take 20 minutes to do a measurement. Even a spot assimilation measurement takes quite a bit of time. So the breeders, quite rightly, to use the me mate, nobody got time for that. Certainly that's not going to be a breeding tool to identify material for crossing. So really photosynthetic improvement has not been a target, mainly for this reason. But that being said, the ACI curve is a really instructive tool for being able to tap the components of the biochemical efficiencies of photosynthesis in crops. And this is because when we combine that with a Farquhar-von-Cameron-Berry model, we can extract both the maximum capacity of electron transport by looking at the high CO2 rate of photosynthesis, and we can get the amount of Rubisco with a Rubisco capacity, VC max, from the initial slope or the Rubisco limited part of the curve. So VC max and J are robust model parameters, and the question is, can we use those to exploit or screen for genetic variation in photosynthetic capacity? And are these model parameters better than spot assimilation measurements? And teleologically, one would expect so, because stomatal conductance can vary quite a lot, particularly in field measurements, and this can be a major confounding influence on spot assimilation measurements. So ourselves and various others have looked at photosynthetic variation, both with the model parameters and by spot assimilation measurements across the large germplasm panels of both wheat and rice. And there's a huge amount of photosynthetic variation in our for the VC max and J parameters in our cereal crops. Even in elite material in wheat, we see about a 30% variation in both of these parameters. When we go into land races and wider germplasm panels, we see even more than that, and certainly even more than that in the IRI panel of global diversity in rice. So there is a lot of phenotypic variation, but can we breed with that? What's the genetic architecture that underpins that, and how can we figure it out? Well, one of the things that excited me about the wheat work, much of this was done by Veri Silva-Perez, a talented PhD student in the centre. And what she did was to look at these large germplasm panels of wheat, some historic, but mostly material that was reasonably recent, the last 2 decades, and examined variation in the parameter VC max and various other parameters, and in this case in response to nitrogen supply, because as we would expect, Rubisco follows nitrogen in the leaf quite closely, because Rubisco makes up more than 60% of leaf nitrogen. But what I found particularly interesting in her analysis was that while there is a relationship between nitrogen and Rubisco content, particularly at low leaf nitrogen, when we get up to levels that are more relevant to what we'd see in field-grown wheat, there's a huge variation in the amount of Rubisco that's invested out of total leaf nitrogen. So the plants have quite a bit of decision-making to go on in terms of how they put their leaf nitrogen into photosynthetic proteins and what else they can do with it. So the question is, can we exploit this? This actually overcomes some of the criticisms of photosynthetic improvement, because it's not just another green revolution trait where you need to throw more fertilizer on. We can actually convince plants to put more nitrogen into Rubisco and electron transport as a percentage of their total nitrogen. And once again, this variation was about 30%, pretty similar to what we saw in our more global analysis. So the question is, can we measure this quickly? So no one wants to do 20-minute measurements per line and look at thousands of different. You would be here, we're dead really to use traditional LICOR for ACI curves. So Viri developed a machine learning procedure where she could look, inspired by the work of Sean Serban in trees. Viri decided that she'd look at whether she could develop machine learning algorithms to examine the light that's reflected from the canopy, from the wheat leaves, and predict photosynthetic parameters by modeling those changes in differences, if you like, between genotypes in reflected light. So by doing that, she came up with some very good correlations, this allows us then to look at large collections of germplasm with these models, and it only takes 20 seconds to get a leaf reflectance measurement, which gives us 6 different photosynthetic related traits, which is a great solution to our problem. And the question, of course, is this photosynthetic variation controlled by genetics or is it by environment? If it's by genetics, we can breed with it. And the way that geneticists measure this is by heritability. So heritability is a number between 0 and 1. The phenotypic variation in the parameter is completely genetically controlled. 0, it's completely environmentally controlled. So as you can see here, a range of our parameters that we can extract with our leaf reflectance. Many of these are highly genetically related, and we can use them to find the genetic architecture, and we can breed with them. So we can pass germplasm straight to breeders and they can use it and be confident that they'll get improved photosynthesis. So we can also then find the genetic architecture underpinning these traits, and we've found a number of really strong loci in these resequenced and highly genotyped wheat panels, and for all of our traits. But interestingly, many of these traits also seem to be associated with flowering time, and I'll come to that in a moment. But how can we use this kind of approach, particularly the genome-phenome approach? Well, globally, we have these large collections of global plant diversity. Many are crops and wild relatives, and many of these germplasm collections have Illumina sequencing systems running day and night, producing massive data sets of gene sequence. So I guess I see the future where the high density, high throughput genotyping and resequence information can be used by us photosynthesis experts to look for SNPs, for single nucleotide polymorphisms across a range of candidate genes in photosynthesis, using our modeling and our biochemistry expertise to look for allele mimics in a common genetic background, and then we can feed those straight into the breeding system. And recent papers suggest that we can deal with not just one gene, but we can do 15 alleles and get a wheat variety in a year by deploying this what's called multiplex CRISPR-Cas gene editing. So I'm more confident now that we can address these things with our photosynthetic capacity. So what controls photosynthetic capacity? It's a bit disappointing in a way when we've looked at some of the genes that are under these genomic areas that control photosynthetic performance. Very few of our structural enzymes have come up, but lots of transcription factors do. And one of the transcription factors that seems to be involved is a transcription factor involved in cytokinin sensing, which is golden-2 or gluck. And we played around with this in C4 rice, overexpressing it to try and get more chloroplasts in the bundle sheath compartment. But most recently, some material we generated has been taken out to field trial by collaborators in China, and by overexpressing this transcription factor, it increases the amount of chloroplasts within the mesophyll cells, but most importantly, it produces ectopic chloroplasts in tissues that don't normally make them. So the reproductive tissue, like the lemma and paley of the seed, produced lots of chloroplasts, and unexpectedly, really, we saw, well, these researchers using that material saw about a 20% increase in yield. And there are a number of reasons for that, but part of that was an increase in photosynthetic capacity in non-leaf tissue, which just goes to show you can't just look at a flag leaf when you generate a transgenic plant. One of the other transcription factors that really attracts me is this NFY group of transcription factors. And I came across this kind of by accident. A group in Japan, Adachi et al., had been working for a long time on something they call the CAR8 locus, which appeared to be controlling photosynthetic capacity in rice. they did some really elegant work over about 6 to 8 years, and they produced near isogenic lines and mapped the gene and the polymorphism, and it's in one of these transcription factors that controls duration of vegetative phase, flowering, abiotic stress, a whole range of different things. But it also seems to control photosynthetic capacity for a given leaf nitrogen, which is intriguing. And when I searched the databases with this transcription factor, it turns out it had been overexpressed in wheat for an entirely different reason. And the transcriptional analysis showed that our whole suite of enzymes that we've got as targets in our photosynthetic improvement, Rubisco small subunit, SBPH, LHC2, P700, cytochrome B6F, they were all being controlled by this transcription factor. So I think there's an intimate relationship between these transcription factors, flowering time, duration, crop development, and photosynthetic capacity. And pulling these apart is quite difficult. And sometimes I think we can throw the baby out with the bathwater by taking our germplasm panels and throwing out the ones that have different flowering times because it makes mapping more difficult. It may be that flowering time and photosynthetic capacity are intimately linked. So I'm pretty much done. You know, going back to my Bruegel painting, can we go fast enough to get to where we need to be without drawing that graph? I'd like to say that I think that by bringing together genomics, phenomics, and synthetic biology, I think we can get to where we need to go. But we've got to be like the guys on the left, beavering away, not the guy having to sleep on the right and thinking, well, we've got 50 years to sort this out. And that's the message for our funding agencies as well, I suppose. So I'd like to close with an acknowledgement slide, which basically acknowledges a whole raft of people, and some people aren't on there as photographs, but huge number of talented people in the centre who contributed to this, and talented and fantastic international collaborators. So I'll finish there. Thank you.
That was very nice. Thank you, Bob. If anyone has any questions they'd like me to put to Bob, please put them in the question box. I will endeavour to put them to him. Otherwise, raise your hand in the question chat thing and I'll try and open your microphone. You were talking about explaining to breeders that you need to increase photosynthesis in the breeding programs. How do you find they respond to that? Because do you find quite often they will say, well, actually, that isn't my top priority because I've got all these other biotic and abiotic stressors that supersede that? Do you find that as a response or?
Well, to a degree. I mean, I guess if you talk to the biotic stress guys, they're always looking for the next resistance gene. You know, you've only got to say UG99 and all the fungal pathogen guys, you know, can get millions of dollars straight away to find novel resistance genes. But, you know, I guess you have to be balanced about it. It's like the solutions to mitigating climate change. You know, you need a distributed solution. So yes, biotic stresses are important, abiotic stresses are important, but the fact of the matter is if you can improve yield potential, then those things kind of sit to depress yield potential below the maximum. So by pushing up yield potential, you can improve crop yields under stressed and unstressed conditions, and that's been shown in the case of drought tolerance, for example. So I guess I am surprised that it's become more of a high-profile target for the breeders because, quite honestly, we had limited success back in the late 80s, early 90s in convincing breeders that this was a viable target. But yeah, the things have gone 180 degrees.
Yeah. And do you find there's ever potential downsides? For example, we had a speaker a couple of months ago, Wenbin Zhu, who was talking about one of the downsides of increasing photosynthesis was a lot of lodging because the head weight was a lot bigger. But I'm presuming if you haven't got sufficient sink capacity, you've also got potential pest problems because you've got higher amounts of sugars. Is that true? Are they things you ever see in these improved crops or not?
I don't think we've got enough information yet. One thing I like to say about source and sink, you know, it's a real bane of my life. I've worked at both ends of the spectrum, and that we've had historically a very unproductive kind of conversation between people who say, well, you know, crops are sink limited and crops are source limited. And the answer to that is it depends on what stage of development you're looking at, you know. But one of the things that does encourage me is that we can see from high CO2 fertilization experiments that there are examples where elevating photosynthesis in CO2 doesn't produce a higher yield because of sink limitation, but it's highly genotype dependent. So, for instance, in rice, that was the first crop where breeders realized they were becoming source limited because they serendipitously produced some rice varieties that had 30% more florets on the panicle. And it was a serendipitous mutation in cytokinin oxidase. So suddenly, rather than breeding progressively for more source capacity in conjunction with more sink, they suddenly had plants with 30% more sink. So they had 30% of the florets didn't fill and weren't fertile. In fact, the carbon got diluted and they got lower yields in those lines than they had in their previous parents. So that's where the photosynthetic improvement stuff came from in rice. In wheat, I think there was a much slower progress because breeders were moving lines into their programs that had both improved photosynthesis serendipitously and improved sink strength through grain number. So it's only more recently in wheat that we've been able to see that photosynthetic limitation.
Okay, we've got someone raising a hand. Christina, can you unmute Menji Fan to ask a question?
They're not connected to audio, so I think they'll have to type it.
Oh, I'm sorry, you'll have to type your question then, please, Menji. Are there any other questions that anyone would like to put through? And please type them in the box if it's not possible to unmute you. I have a question in the meantime. Does improving photosynthetic efficiency always mean increased resource use efficiency? I mean, when you talk about increasing, for example, SBBAS and Rubisco amounts, are they necessarily increasing resource use efficiency? Or do you think we haven't got enough information on those yet to be able to say that? Because I'm thinking they will require more nitrogen and resources.
Well, I don't think we've got enough, we don't have enough work done yet in our major crop species to know for sure. But what encouraged me was that graph of leaf nitrogen versus VC max. And, you know, we thought that really that would be a pretty good straight line and everything would fall on the straight line. But this variation, large variation at a given leaf nitrogen for how much nitrogen is invested in Rubisco and electron transport components suggests to me that plants have quite a lot of flexibility in that. It's just we don't really understand how that works. And when we plumb the literature, there's pitiful information. You know, nitrogen use efficiency has been a vexed topic in crop physiology for many years, and our understanding of how a plant decides where to invest its nitrogen is even less well understood. So yeah, I think we just don't know. And it's obviously very important because of the pollution coming from the inappropriate application of nitrogen. And of course, in the UK, that's a very hot topic.
I've a question here from Tracy Lawson from Essex. What about the potential to increase photosynthesis in non-foliar tissue? Could this make a contribution to yield?
Yeah, definitely. And I reckon, you know, this material that we produced in C4 rice, when we measured the photosynthetic rates in leaves, just under standard conditions, they weren't spectacularly different, even though there were more chloroplasts per leaf area. So we really didn't think that there was much value in this as an approach. But the material was passed on to colleagues in China, and as I say, they field trialed this and got this quite large increase in yield. And then another group had a look at what these transcription factors were doing, and yeah, the panicles were way greener, so the lemma and pala were doing more photosynthesis. The stems were greener, the leaf sheaths were greener, and they stayed greener for longer. So yeah, I definitely think that non-foliar photosynthesis can make a contribution and possibly even more so under stress conditions. So, for instance, in wheat, under drought stress, up to 30% of grain carbon can come from ear photosynthesis. So, yeah, boosting reproductive structure photosynthesis, I think, is a really worthwhile topic, and I know that Tracy's very interested in this.
Okay, I've got one more question here from Anil Kumar Aruji. Asked whether there's any variation in photosynthesis in wild cultivars of crop species.
Yeah, definitely, even more than there is in these elite germplasm sets. So in Viri's work with wheat, she looked at some wild relatives and some land races, and yeah, they had huge variation. But the difficulty in looking at this really wild germplasm, in particular some of the material that comes from these global germplasm banks, it's so different, you know. So its flowering times are all over the place. It even looks completely different. You know, you saw that image of the global diversity set for rice. They just have so many differences in terms of morphology, tillering, flowering time, duration. So to compare apples and apples is quite difficult. But the initial screening that was done on the wheat germplasm would suggest that yes, there's really good novel sources of genetics from wild progenitors and wild relatives, and we have a capacity now to bring that allelic variation into elite lines without years and years of back crossing to get rid of all the lousy stuff that you don't want from those wild relatives. So yes, the answer is yes and yes.
And I've got another question from Menji Fan. Does higher sink capacity have a positive effect on photosynthesis?
Yeah, it can do. You know, a lot of the bad press for photosynthesis came from early work where people were cutting off ears and seed heads and looking at what effect it had on photosynthesis. So sugar feedback on photosynthesis is a well-known phenomenon, and particularly pertinent under high CO2 enrichment. So yes, you definitely need to have high sink capacity to take advantage of improvements in photosynthesis, or you do get feedback. But there are really interesting mechanisms. When the cereal crops, for instance, are making the ear inside the boot of a wheat plant, they sense how much sugar is coming from the leaves, and they adjust the size of the ear according to the supply of carbon. So it's almost like they're trying to have a prediction of what the season's going to be like. And so if you boost photosynthesis, often you'll get an increase in the size of the sink to match. But of course, everything has its limits. You move a limitation from one thing to another. So yeah, you need a good balance between the two.
I've got a last question here from Christine Rains asking, in addition to non-foliar photosynthesis, there's also differences in leaves at different stages of development. They're not only full expanded that we normally focus on. I presume you agree with that?
Yeah, absolutely. And Marsha might touch on this. You know, I think it's really dangerous just to look at one leaf at one stage of development. And we saw this with our wheat, most recent wheat work, much of which is yet to be published. We looked at tillering, booting, flowering, and 4 days after flowering. And, you know, ranking of genotypes was actually quite different at those stages, and also in particular heritability of the photosynthetic traits. So yeah, we need to take a holistic sort of view of crop photosynthesis, and I think that's where we came unstuck early on, you know, that people said, oh, there's no relationship between flag leaf photosynthetic rate and grain yield in wheat. Well, they're probably right, you know, if you measure it at a particular time. But if we have an integrated approach, which of course is more experimentally difficult, we'll probably get more accurate answers.
Okay, well, we're going to have to move on from that. Thanks very much. And I'm delighted now to welcome our second speaker, Dr. Maria Ermakova, who is the lecturer at Monash University in Melbourne. She paved a way to genetically redesign plants for more efficient photosynthesis and higher productivity, and was subsequently awarded the Peter Goldacre Medal from the Australian Society of Plant Scientists last year in 2022. She leads the Plant Energy and Biotechnology Research Group at Monash, where they're aiming to create more energy efficient plants which are better suited to the Australian climate. She also worked at the ARC Centre of Excellence for Translational Photosynthesis and is part of the C4 RISE project. And today she's going to talk to us about developing strategies for improving C4 photosynthesis. Over to you.
Thank you, Louisa, and thank you so much for inviting me to present my work on improving C4 photosynthesis. Most of this work has been done within the Centre of Excellence Translational Photosynthesis at the Australian National University. Now I continue it in my group and on plant energy and biotechnology in Monash University. So why are we specifically interested in C4 photosynthesis? This is because it's very important for Australia, and on this map you can see that pretty much half of Australia is taken by the grasslands, in which almost 100% of photosynthesis is undertaken by C4 plants. And although C4 plants represent only 5% of terrestrial species, they account for 20% of primary productivity. And because of this, some of the most important grain and energy crops we have are C4 plants. For example, maize, which is in our top 3 of most important crops, together with rice and wheat. And sorghum has been increasingly important for some dry areas in the world. And miscanthus and panicum are some of the dedicated energy crops which used for biofuel production. So why C4 photosynthesis is so much more efficient? It's because it's working as a CO2 pump. And in C3 plant, everything happens in one cell, in the mesophyll cell, where rubisco is constantly exposed to oxygen. And as you know, this leads to a production of 2-phosphoglycolate, which is a toxic compound, which needs to be then regenerated in the photorespiration, which requires additional energy input from the plants, and overall decreasing the yield potential of C3 crops. But in C4 plants, photosynthesis is divided between mesophyll and bundle sheet cells, and rubisco and other enzymes of Calvin cycle are encapsulated in the bundle sheet cells, which are pretty much gas-tight, while CO2 is initially fixed by PEP carboxylase enzyme in the mesophyll cell into C4 carbon compound, which diffuses to bundle sheet cells, where it gets decarboxylated to release CO2 around rubisco. And because PEP carboxylase itself is not sensitive to oxygen, and due to high activity of the C4 cycle, there is in CO2 enriched atmosphere around rubisco in bundle sheet cells, which pretty much eliminates a reaction of rubisco with oxygen and prevents expenses to photorespiration. Setaria viridis recently emerged as a new model for studying C4 photosynthesis. It's a close relative of Setaria italica, which is an important crop in China and India. And it's also a monocot and NADPME type C4 photosynthesis, which makes it a good model for maize, sorghum, and other important C4 crops. And it's also short and easy to manage in the lab and has available transformation technique. And therefore, we wanted to take advantage of this available genetic transformation of Setaria and test various strategies for improving C4 photosynthesis that we identified using the biochemical model. So in the model, the ACI curve of C4 photosynthesis, which is dependence of CO2 assimilation rate from the intercellular CO2, described by this ACI curve is described by 2 equations. So you can see it has these 2 distinct parts. One is the initial slope of the curve, and the second is the saturating part. And according to the model of the C4 photosynthesis, the saturating part of the curve can be limited by rubisco, electron transport, or RGBP regeneration, while the initial slope of the curve can potentially be limited by PEP-C activity or carbonic anhydrase activity. This is the first enzyme of the C4 pathway, which required to hydrate CO2 to bicarbonate to then be fixed by PEP-C. And also mesophyll conductance can limit assimilation in this initial slope of the curve. And 3 of these targets, electron transport, RUBP regeneration, and mesophyll conductance, we tested in Cytherea. So first, I'll tell you about our work on increasing electron transport rate. For this, we used a method that was already known from work of a group of Christine Rains in C3 plants, where they used overexpression of the nuclear encoded risky iron sulfur subunit of cytochrome B6F and showed that plants which overexpressed risky could have increased electron transport rates. And this was shown in Arabidopsis. So we wanted to use the same approach and try to increase risky abundance in Cytherea. And we transformed Cytherea and recovered some plants which had increased risky abundance per leaf area. And we also showed that these plants had increased cytochrome B6F abundance. This was done by separating the thylakoid protein complexes using blue native page, and then blotted these complexes with risky antibody. And we showed that the transgenic plants with risky overexpression have more cytochrome B6F in both mesophyll and bundle sheet cells. And then when we analyzed the electron transport and gas exchange properties of these plants, we discovered that increased risky abundance resulted in higher electron transport rates at high CO2 and high light conditions, which is seen by the increase of efficiency of photosystem 2. And these plants also show the corresponding increase of the CO2 assimilation rate at high CO2 and high light. Therefore, we showed that increasing risky content could accelerate the steady-state electron transport rate in Cytherea, and it was also the first work to show that C4 photosynthesis is limited by electron transport at high light and saturating CO2. And our Cytherea plants overexpressing risky also could grow faster than wild type. And we were interested what would be the effect of increasing risky content then on yield of real crops. And for this, we chosen to overexpress risky in sorghum, which is one of the most important crops, and it's also a multi-purpose crop that's increasingly important for agriculture in the world. For this, we used TX430 variety of sorghum, which is easy to transform. And we obtained 3 independent transgenic lines, which had increased risky content per leaf area. So to our surprise, when we analyzed electron transport and CO2 assimilation in risky overexpressing sorghum, we have not identified any increases in steady-state electron transport assimilation rate. However, we have identified increased electron transport and CO2 assimilation during the photosynthetic induction. This is the process of activation photosynthesis when plants are shifted from dark to light. And you can see that over the time course of 30 minutes, when dark adapted plants were shifted to 1000 microeinsteins, all 3 transgenic lines overexpressing risky could activate their electron transport and CO2 assimilation rate faster than the control plants. And we think that this transient increase of assimilation at the dawn every day actually resulted in these transgenic plants being able to accumulate more biomass. So this is the plant grown in a glass house, and you can see that all 3 transgenic lines were taller than the control plants. They had more leaves and accumulated more biomass by the end of the growth season. And they also produced more seeds, both by number and by weight. So this work showed that although in sorghum cytochrome B6F activity does not seem to limit the steady state electron transport rate, but increasing risky content still could be beneficial for enhancing yield of C4 crops, because it could accelerate the photosynthetic induction. And now when we have a bit more information about the increase in risky content in different C3 and C4 species, we kind of can compare results obtained in different species and see some patterns. And it looks like that when we move from model plants like Arabidopsis and Citeria to more of a model crops like tobacco and sorghum, we really see different effects of increasing risky abundance on electron transport, because it moves from more of a steady state effect of increased electron transport to transient increases, which could potentially be explained by some positive selection for cytochrome B6F abundance during domestication of plants, or maybe it tells us that there are potentially some different effects of photosynthetic control in different species, which prevents potentially positive effect of risky overexpression on electron transport. So this is something that is interesting really to explore further, and something that we need to keep in mind when trying to translate results from one species to another. Now I move to the second part of my talk, which is related to improving mesophyll conductance in Citeria. So mesophyll conductance, or GM, is the ease with which CO2 diffuses between intercellular air spaces and mesophyll cytosol. So here you can see that to get to the mesophyll cytosol, where it's getting fixed by carbonic anhydrase, CO2 needs to cross the cell wall and the plasma membrane of the mesophyll cell. And it's specifically plasma membrane, due to its crowded nature, that is considered pretty much CO2 impermeable. But we know that some transmembrane protein channels called aquaporins, which were initially identified as water pores, also able to transport some small solutes across the membrane, like cations, ions, boron, urea, and also CO2. And we were interested whether there are some CO2 permeable aquaporins which contribute to the CO2 diffusion in leaves of Citeria. And to identify these aquaporins, we first expressed 12 of plasma membrane intrinsic proteins from Citeria italica in yeast. And then those aquaporins which were effectively targeted to the plasma membrane of yeast were assayed for CO2 permeability using the stop flow cytometry. And in two independent experiments, we identified PEEP27 as CO2 permeable, but to our surprise, this aquaporin was not expressed in leaves of Citeria, but in roots. However, we were still interested what happens if we do express this CO2 pore in leaves of Citeria, and we created transgenic plants which express PEEP27 in fusion with FLAGTAC. And we confirmed that 3 independent transgenic lines expressed this protein in leaves. And we also confirmed using immunolocalization on leaf cross sections that this aquaporin was efficiently targeted to the plasma membrane of mesophyll cells. So it was in a right position there to be able to act as a CO2 pore inside the leaves. And indeed, when we measured the mesophyll conductance on these transgenic plants, which we measured using the oxygen isotope discrimination, we could show that 2 of our transgenic lines had increased CO2 diffusion within the leaves. Next, we analyzed gas exchange properties of these transgenic plants and paid specific attention to the initial slope of the ACI curve. And by doing this, we could show that all 3 transgenic lines expressing the CO2 pores in mesophyll had also elevated initial slope of the ACI curves. And when we plot initial slope of ACI curve versus mesophyll conductance, all 3 transgenic lines have increase of both, and this increase is following the prediction of the biochemical model of C4 photosynthesis. Therefore, we showed that increasing engineering mesophyll plasma membranes with CO2 permeable aquaporins could increase mesophyll conductance in Citeria. And by this, we also showed that mesophyll conductance indeed limits the C4 photosynthesis at low CO2. And I'll briefly tell you about our recent work on trying to stimulate RUBP regeneration of Citeria. For this work, again, we used the method available from C3 plants for stimulating RUBP regeneration via overexpression of SBBAS. And there was a lot of success from the group of Kristin Rains and other groups, which showed that increasing SBBAS abundance in C3 plants led to increased CO2 assimilation, increased biomass and yield of many of C3 plants and crops. And this is because SBBAS is one of the first enzymes of the regenerating part of the Calvin cycle, which kind of controls a lot carbon flow in C3 plants, because it sits here next to the branching of using triose phosphates for sucrose or starch synthesis, or deciding whether they become regenerated into RUBP. So we wanted to use the same approach and overexpress SBBAS in Citeria, and we received several transgenic lines which had quite significantly increased content of SBBAS in leaves. And we also confirmed that this extra SBBAS was in bundle sheet cells, where it should be acting in Calvin cycle, because most of the transcript that belongs to Brachypodium SBBAS that we overexpressed was found in the bundle sheet cells. But increasing content of SBBAS did not show any effects on plant biomass. And also, when we tested CO2 assimilation in these transgenic plants, we used all kind of conditions, different light intensities, different CO2 and different temperatures, but we could not find any effect of increased SBBAS content on assimilation rate. And when we did plot maximum CO2 assimilation rate versus relative SBBAS content, even though we reached quite significant increases in SBBAS content, there was absolutely no effect on CO2 assimilation. And therefore, we could show that SBBAS content does not limit C4 photosynthesis in Citeria, although it could be maybe a species-specific effect. But there are some properties of C4 plants that confirm this conclusion. For example, the abundance of SBP, which is the substrate of SBBAS, has been decreasing during the evolutionary transition from C3 to C4 plants in genus Flavaria, which probably indicates that there was some increase of SBBAS content or activity in C4 plants over the evolution. And also, most likely, because in C4 plants, the starch and sucrose synthesis is quite more complicated than in C3, because it's separated between different cell types. It's also unlikely that SBBAS would have the same control over the carbon flux as in C3 plants. So coming back to the targets that we identified initially for improving of C4 photosynthesis. Now, our works and works from other groups, like from Stern group, showed that Rubisco and electron transport indeed have effect on the saturating part of the ACI curve, and increasing amount of Rubisco or increasing the electron transport rate could stimulate photosynthesis at high CO2 and high light. While mesophyll conductance, we showed that stimulates initial slope of the ACI curve, but we still don't know whether there are any effects of increased PEPC activity or carbonic anhydrase activity on initial slope. And here is some of the works that I'm gonna continue at Monash University. And what we want is to move more to testing the successful targets in sorghum, and also to start testing this successful traits in elite lines and in the field experiments to see like whether all these traits will be really helping us to improve yields of real crops. And also we'll continue developing and testing new strategies for improving C4 photosynthesis. And for this, we're already looking at how improving PEP-C catalysis will affect C4 photosynthesis in collaboration with Rob Sharwood from Western Sydney University. And we're also working on improving the model of C4 photosynthesis to have a bit more detailed description of electron transport in 2 cell types, because this will allow us to find new targets for improving photosynthesis, potentially by more fine-tuning of electron transport in different cells. And with this, I would like to thank you for tuning in today, and also thank everyone involved in the work that I presented. Of course, Bob and Susanna, and many people from Bob and Susanna's lab. And of course, group of Christine from University of Essex and our collaborators, and also Australian Research Council, Monash University, and Australian Academy of Science for funding. Thank you very much.
Thank you very much, Maria. That was really interesting. Some beautiful graphs. I've got 2 questions already here. Please keep your questions coming. I think they're asking a similar thing. One's from Zini Johnson, and the other one is from Elisabetta Karma Silva, and they're asking whether you grew your sorghum plants under fluctuating light, and was the biomass production observed just under constant growing light. I think they're talking about the risque overexpressor sorghum plants, where they grow better in the field or in the lab.
Yes, so the experiments that I demonstrated were done in the glasshouse with the natural light that we have here over the summer in Australia. So it's quite high light and kind of pretty much mimicking the light conditions in the field. And yes, we've seen an increase in these conditions, and we can't really say what we attribute this increase specifically. We definitely know that induction of photosynthesis is increased, but it's also possible that some transit changes in electron transport during like this fluctuation of the light during the day can have potential effect, also contributing to the increase of the biomass and yield that we see. But that's something that is quite hard to test, because that would require growing these plants under constant light, and it's not very easy for sorghum because it's a big crop, and we really could not find a way to grow these plants in growth chambers with the constant light conditions.
I have a question from Felix Bouchet from the University of Munster. He says, thank you for the talk, great talk. Do other B6F subunits also accumulate more in the risque overexpressor lines?
What we could see is that there is more of the whole assembled complex in the thylakoid membrane, and we can see it both by separating complexes with blue native page and testing abundance of the whole B6F complex. And we also showed it in tobacco by measuring activity of the complex via the cytochrome redox assay. So, but it's also on isolated thylakoid membranes. So yeah, presumably there is more of other subunits as well, or at least more subunits that finally contribute to the assembly of the full complex.
He also asks, given the entanglement of state transitions with B6F chlorofluorescence measurements, did you check for state transition phenotypes in the OX lines?
It's not something that we measured specifically, but yeah, there is definitely some effect on state transitions. We also seen it in tobacco because we can see different effects of far red and red actinic light, and yeah, but that's something that we probably we're interested to study more.
Okay, thank you. Michael Mesler asks, did you check the chloroplast ultrastructure of your overexpressing SBBAS and risque lines in comparison to the wild type?
No, unfortunately, we haven't tested that. And yeah, there are possibly some changes with Riskia. At least based on the Blue Native page, we could see that there were no any drastic effects on abundance of other photosynthetic complexes, including like PS2, LHC2 super complexes. So maybe we don't expect a lot of changes, but yeah, we haven't really checked it.
Would I be right in thinking that your work on these overexpressing SBBAS and risque proteins, are they both in NADP-ME types then of C4 plants?
Yeah, this work was done in Sidera, which is NADP-ME type, yeah.
And did you check, has it been done in anything like the NADME or the PepCK C4 types? Because would you expect a difference? I think in your paper you argue that you've got low PS2 activity in NADP-ME types, is that right? So I just wondered in something like Amaranthus, which is an NADME, might you expect the same thing to happen? Or
yeah, that's an interesting part. We would be interested to see what effects of all these improvements in different subtypes, but I guess it's not very easy to transform any model plants in other subtypes. Like regarding Riskia, this probably will be quite similar because I think all subtypes of C4 photosynthesis are kind of limited by electron transport, and this just comes from general activity of the C4 cycle as CO2 concentrating mechanism, which kind of shifts the limitation more to supply of NADPH and ATP from the electron transport. But SBBAS wise, there might be some differences because yeah, the subtypes might have quite different regulation of sucrose and starch synthesis, and also I don't know how much. So one of the complications of NADP-ME type is that part of the PGA from the Calvin cycle in bundle sheet cells needs to go to mesophyll to be reduced to triose phosphate, because bundle sheets do not produce their own NADPH, or not as much. So yeah, this will be also quite different in other subtypes. So potentially, yeah, SBBAs might have maybe more control over the flux there.
Has anyone ever looked at the difference between efficiencies in the three subtypes then? Is there a number that says which is the greatest photosynthetic efficiency? Maybe. I mean, I think crop plants are NAD. Is that right? Most crop plants are MADP-ME types, sorghum, maize.
Yeah. I mean, NADP-ME is considered probably the most efficient, like the highest quantum efficiency of other subtypes, but I think NADME and NADP-ME quite close to each other. But it might be then there is more room for improvement in other subtypes.
Go on, Bob.
Can I make a comment, Louisa, on that? Back in 1990, I modelled that, and the most efficient subtype should be a pepcarboxykinase. But it turns out there never is a pure pepcarboxykinase. It's always mixed with other subtypes, so you can't demonstrate any difference in photosynthetic efficiency experimentally between the PepCKs and the other types. So, yeah, I think Marsha's right that it's a bit of a smoking gun that most of the domesticated C4s, apart from switchgrass, are NADP type, so it's got to have something going for it. But you know, even NADP types are mixed. You know, they can have a bit of PepCK, or they can need a bit of aspartate. So maybe, you know, they just have a nice bit of flexibility and are more easily domesticated. But you know, Amaranthus has got something going for it. It's the Indian subcontinent seems to like it, Louisa.
Well, always keep my eye on Amaranthus for me. Right, okay. I've got a last question here. Is it possible to increase photosynthetic efficiency of tree species? What will be the challenges in it?
It's probably possible if we can ever test it in tree species. Not sure if there are any.
Are there any species? I don't know. I presume there isn't any.
It's one. I think there's one. Rowan Sage told me that there's one, maybe, but there's not. No, there aren't many that I'm aware of, and it possibly, I don't know. People say that the efficiency of C4 at low light's not great, so you know, to get a tree to grow up through the canopy, it's at a disadvantage when it's at a seedling stage. Maybe, I don't know.
Yeah. Okay, well, I think that's all our questions, and it just leaves me to thank you very much for what was a very interesting discussion, and it was great to have some C4 work in the webinars. Really enjoyed it. So yeah, I'd like to thank both our speakers. It was lovely to hear from you. Thank you for staying up so late for us. I know it must be about 9 o'clock at night now there. And reminds me to ask you to register for next month's webinar series, which is with Sebastião Capabauca, who is talking about aquatic rubiscokinetics, and René Klein-Langhorst, who is talking about the Crop Booster P project. So it should be a really good webinar. The registration is open for the next 2 webinars, so go ahead and register. And if you've missed any of the webinars, they are many of them are recorded, as well as our workshops on genotyping, phenotyping, and field trials. So feel free to access them on the website, and of course let us know what you think if you like. We're always interested in your feedback about the webinars and about the workshops. So that's all from us, and thank you very much from the UK and from Australia. Goodbye.